Thank you, Lane.
No problem.
And thanks to the organizers for this entire meeting. It's so exciting to be back, and I'm really honored to give this talk. And what I did when y'all asked me was I went into my office where I have a box that says Iowa. And it goes with me wherever I go. I mean, from office to office. And I haven't looked in it and for a very long time. So I opened that box, and in it were boxes of slides from my collection back in the day, and a whole bunch of notebooks, including my notebook from the '89 Reimer class, and then a bunch of correspondences from back in the day where you write letters to people with a typewriter, okay? And so it was like a real treasure trove, and it made me feel really old. And I think I might be older than Rex. After I was going through all that, I'm pretty sure I am. But I wanted to start with this picture. This is from the NADS that I hosted down. What was something happened? Something down. Wait a minute. The NADS I hosted down on Isla in 2003. And the reason why I brought this picture up is that it has some people in it who, this might have been their last NADS or next to the last NADS or something. People who are still with us in all the ways that Sarah mentioned. And so we see Charlie there, and I think that was his last NADS. We see Sam Van Landingham, who came to that NADS with a wooden suitcase with his Shakespearean outfit that he ended up wearing down to Fantasy Fest. And there's Stormer, and we got him to wear a dress. Dick Cruzum, too, we put him in a dress. And there's John and Sarah Kingston, who we almost lost to the scuba dive or the snorkeling trip, and Norman. I don't remember getting him into a dress. And we drove everybody down in the university van to to Key West, where the Halloween Fantasy Fest was going on, and it was like super, super fun. And I only got in trouble like 5 years later, and it was after I got tenure, and it wasn't even for driving the university vehicle down there. That's another story for later on. Anyway, what I wanted to talk about was sort of the topic that everybody's been talking about today, which is that we're all connected to each other in a lot of different ways. We're connected to each other through the places, including this place that so many of us have held quite dear for many, many years, for the training and support that we've received here. And through our study organism that itself is very connected to other organisms in ways that we've learned about through each other and through Lakeside Lab. And so I wanted to start with the people part of this. And with my own little story, I was an undergrad at Kent State University, and my professor of limnology was Denny Cook, and he had done his PhD work on Lake West Okoboji. And when I was consulting with him about where to do my master's, he said, you really ought to check out Iowa State and Roger Bachman. I wanted to go into limnology, and so he lined me up with Bachman, and Bachman said, before you start your grad program, go out to Lakeside Lab and take the limnology course taught by Dick Boberg, who was the director at the time. And so I came out a little early, 2 weeks before that class was supposed to start, and they put me upstairs with all the girls at the time above the dining hall, which did not look like this. It looked like a barn at the time. And there was this huge upstairs as big as this tent with like 20 bunk beds. It was super fun. But it was there that I found out the limnology class was full and they had no room for me. And that I was going to have to take this other class called the Ecology and Systematics of Diatoms. And I got so lucky. It was the last year that Charlie taught the class. It was 1989. And here's our little class. I must have taken this picture. It was in the box. And Julia Christensen, where are you? You were with us. She wasn't enrolled in the class because she had had it 10 years before or something. But she came and she was the one thing I remember about you, Julia, is that you had the slide, a few slides that you brought with you. And there were so many species on your slide that it took you the whole entire summer. You had to count until you tapered off with your rarefaction curve, and you just never reached your asymptote. You just never reached it. And you were like, every day it would just go up and up, and you would show it to us. And I remember thinking, oh God, what am I getting myself into? Are all the slides like that? So did you ever finish counting that slide? That was awesome. And then Lauren Hatch, she was kind of a rascal. He ended up over in California doing limnology. And James Baker, he was the son of Dick Baker, who taught the geology class. And James ran the kitchen crew, and he knew how to get to the ice cream any time of day. So that was like a real benefit. And James is now a neuroscientist at University of Miami, and we get to see each other sometimes. And this guy, and I don't remember his name, but he worked for the DNR and he learned his diatoms with us. So that was our class. We had so much fun. We did the usual things. We went out and collected from day one. And Charlie, this is Charlie out there. He's got his pipe. He always had his pipe in the field. Most of the time it would drop into his waders. But Jackie really tried to help him with that by sewing little holsters for his pipe into his waders. And I was trying to think of some stories. The one that comes to mind most clearly is when we had the 15 passenger van and we were driving it. He would always drive us around in it. And he was really obsessed with collecting in ditches. We always had to stop at like any ditch that had water in it. And he would pull way off the side of the road, like I don't know why, because nobody's on these roads. And we would get out, we'd collect the ditch and get back in. Well, one time we're way out in the middle of nowhere and we pull off into this ditch and he pulls way off and the car is like tilting to the left like it's gonna fall over. And he said, get out, but not out the door. He said, climb out the windows on the right. And we're screaming and everything. And meanwhile, he's running down the road and he sees this farmer on a tractor who was nice enough to come down and tow the van out of that ditch. And Charlie was so grateful. He had this $20 bill and he's running along after this tractor trying to pay this guy, and the farmer's refusing to take the money. And then we went straight from there to Little Miller's Bay, where we hung out all the time. But this was like a little earlier in the day than normal to have a pitcher of beer. That was like the usual thing. That was our class t-shirt, and we made it to honor Charlie that year, and it said Iowa Lakeside Lab, and it's the shirt now that you associate with the whole lab. Lauren made the diatom, and I made the little heron, and James made the crayfish, and James's sister made the Chris made the salamander. Anyway, it's fun to see that. And then this was the porch people. We were always hanging out on the porch. Early 90s. I'm not sure if this was 89 or early 90s because Stormer's here. And there's Jackie and there's Charlie and there's Dave Zarnicki. And Dave would come visit us and he would have his big cowboy hat on. And he would always walk very deliberately into the room and he would say, what it is. And he would hover over you at the microscope, what it is. And I'm like, I don't know what it is. I don't know what it is. But what is it? You know, that's why you're here. He would say what it was, and then Charlie would come by and say it was something else, and Reimer would say something else. Oh God, what am I getting myself into here? Anyway, it was good times, and here's some more pictures from early 90s, and like lots of you, you know, we get attached to this place and we keep coming back, and I kept coming back through my master's and PhD work, because I was really working with major professors in other disciplines. And so Charlie helped me a ton, and Stormer helped me a ton. And that's you, and then your birthday suit, I think, a hot day. And Charlie and Donor, I think, and Jen, and Stormer showing us how to use a plankton net appropriately. And here's Jen Slate looking like she's trying to pull Charlie out of a ditch, like not an uncommon thing to do. And so it was fun.
Did you have to pull him out of the fence?
Yeah, probably. I'll talk about Excelsior Fen in a minute. Yeah, I mean, it was always like jumping around out there. You did?
Yeah, we did.
Well, the nice thing, Charlie was always inviting us up to the academy, right? Super invitational to get these little travel grants. And that was important again for me because I didn't have the whole library around me and stuff. So I would go up there and then he would write letters. He was really great at writing letters on his little black typewriter. And I found those in that box of treasures. And this one's hilarious. It's like about amphora slide I sent him or something. And then at the end, I hope it's okay, Jackie, it says, the 3 young ladies at home, that's you and the kids. I'm so glad you're here. And the 2 old ladies here at work, which I think is Sue, who did all the illustrations, and probably Ruth Patrick, maybe, I don't know, are fine. I always thought I'd like to have a harem of 5, but certainly none of these 5 qualify at all. Give you a little flavor of Charlie. I couldn't believe it when I found this. I'm like, this is great. And this is the only picture. So thank you, Jackie, for hosting me all those years. And you got to stay in their wonderful finished basement around all the diatom books of the world, you know. And then this is my main picture of everybody doing hula hoop. I found all these pictures, they were printed out, you know, it's before digital. Anyway, we're attached to each other. We're attached to this incredible place, McBride Hall, and the 3.5 drink kettle hole. Who doesn't love it? I wrote my thesis in this little room here and hid from tornadoes in this little room there. And Excelsior Fen, one of my favorite places, and that's the first place that I ever collected a paraphyton mat, and I'll never forget it, a big clump of calcareous paraphyton. And in it was this diatom, a little Masticloia, Gravellia of some sort, maybe. And I found my notebook. We had to identify 40 diatoms. And I have this list here. Some of them have X's. Maybe I got them wrong. I don't know. And it's a list of taxa C and ID. And then this is like notes from the field, and it says Roman numeral 7, like July 19th, 1989, Excelsior Fen, pool in Fen, and that was where I got that little piece of paraphyton, and it had Masticloia gravellii, maybe, and Masticloia smithii, a variety lacustris that ended up becoming a very good friend of mine, I thought, in the Everglades. And then down here is another sample we took there in a cow hoof print, which was another obsession of Charlie's. He loved sampling. If you saw a cow hoof print, you had to sample it. I also found this wonderful, he would take Houston stuff and translate it and give it us little printouts of it. And this is wonderful. It talks about ways in which you need to think twice about your relative abundance measurements and rarity and commonness and frequency of diatoms and what it actually means. And at the end, it says microenvironments in any given region must be carefully explored because that's what matters the most to diatoms. And that's where we learned, you know, think like a diatom. And then I found our final exam. It was 4 pages. And we were actually given a whole day to study for it. And we went out on the pontoon, it was called study hall. And we tied it out and we sat there and with flashcards and studied all day. And this exam was so dang hard. I think that more people would flunk it than flunk the certification thing we did in the Georgia meeting, right? Because it was hard. And I look back at this exam and I'm like, wow. And I looked at my answers, some of which are here, like to this dichotomous key we were supposed to make. And I'm like, what the hell was I doing? It was really bad. And then the last question is awesome. It says, and I'm going to use it, all those who wish to help with the exam writing can do so here. Write 3 reasonable questions on diatoms, which A, you think everyone in the class could answer, B, you think only some in the class could answer, and C, ones you wouldn't expect anyone in the class to answer with you as a possible exception. And I think that's where all these questions on this test came from. And then it says, damn it, is das examen fertig, which is this class is now over. Or this, yeah, exam is now over. And then he never gave us our grades. He just came in and he said, y'all have A's. Nice of him. We had class projects. Mine turned into my thesis. And this is where I first started learning about why microhabitat matters. I studied this little diatom attached that grows attached to daphne and copepods and stuff called Sinedra or Fragillaria cyclopum, and it's really lucky, you know, it attaches to the thing that eats it, so that's smart. And then as this guy's swimming around and pooping, you know, it gets all that juice, which is important because it mainly lives in oligotrophic lakes. So these 3 ways that we connect to each other and to the work that we do and to the diatoms that we study who are themselves connected to other diatoms is really important, and it's powerful and it's rare. We are so lucky. And it's mattered a lot to my laboratory. And I just wanted to do a couple of highlights of how I think the ways that I was trained here and the ways that my students continue to be trained so much by all of you, it matters to the work that we're doing. And if I have a minute, you know, we're studying persistent attachments in the Everglades. We study these periphyte mats that look a lot like the ones in Excelsior Bend. And here's some videos that Franco Tobias, my lab manager, took showing how we get around out there by helicopter and by airboat. It's a lot of fun, by marine boat, and sample periphyte in these different habitats. And just a couple of snapshots, and you've heard a lot about these. These are sort of recent updates from the lab. And Paige this morning taught you about how she's learning that plant biomass is just as substantial as periphyte biomass in the Everglades, and that being around periphyte and diatoms for a plant is beneficial. Growing in that attached growth form matters to your persistence during the dry season. Tommy Shannon, who couldn't be here because he's writing his dissertation but misses everyone, and we'll have to call him or something because I feel sad for him. He got a scholarship that bought him this incredibly fancy high resolution underwater camera that you can't really see this, but I'd love to show you in a dark room or something how clear these pictures are. This is going through the water's surface, and you can see individual clusters of diatoms and single strands of cytonema, cyanobacteria in these pictures. And so we're digging into that community that's highly interactive in these maps, looking at the diatoms, the other algae and cyanobacteria, the crystals of calcium carbonate, all this extracellular polysaccharide that there's exuding out. You can see the mucilage forming around these stems. And with the diatoms, We've been sampling 150 sites throughout South Florida on an annual basis for more than 20 years now. So we have some incredible data sets. Franco Tobias has been with me that whole time, and he's been counting and identifying everything from this project. It means I have no observer bias in it. It's incredible. It also means he's counted 1.5 million diatoms we figured recently. And so I'm glad he doesn't have the rarefaction curve that you did, Juliana. It would be several lifetimes of work. No, we have a very species poor flora in the Everglades that's easy to count relatively, and it's very responsive to changes in hydrology and nutrients that are happening. And we can provide these kinds of reports, green, yellow, and red, good, medium, and bad, to the Congress that's funding the restoration of the Everglades. But Tommy's doing more interesting stuff with this. He's taking some of our dominant species. These are GIFs showing the changes in their abundance over time, over 20 years. And he's finding that the drivers of those year-to-year changes are different depending on what scale you look at them. And in fact, diatoms are responding to different things at their little tiny scale than it shows up in this whole large spatial scale of the design that we're using for assessment. And so he has a very cool paper that he's writing right now on multi-decadal scale dependent dynamics of this meta community across space, time, and biotic and abatic ecosystem realms. So we've been zooming into this mucilage property of these mats, and I know lots of you are really into mucilage formation and didymo and other communities. What's it there for? What it is? Sylvia Lee started digging into that a little bit, and I couldn't find a cute picture of her and Stormer, so I had to deal with this one. Just kidding. It's like the most adorable picture ever. And Stormer said to us when he saw this diatom we kept finding, and we were calling it Massicolia, Smithii variety lacustris, just like the one at Excelsior Bend, because it sure does look like it and sure does live like it. He said, no, it has really straight sides. You better look a little more closely. So Sylvia did, and she named it Mastigoia calcarea, we et al., with Mark and other people helping. And she zoomed in on it, and it produces mucilage out of its the little breaks in its protectee, these little strands of mucilage that hold it up into the mat. It's super cool that way. And then Katie Stansbury came in after Sylvia and dug into this further. She was able to grow up. She grew up the Mastigoia calcarea in culture, and measured the area of mucilage around the cells. And she was able to find that the cells that were smallest and looked like they were exchanging gametes were most likely to have the biggest halos around them. So that halo seems to be helping with gamete exchange. And we've learned in other work that we've done that it is more abundant in the shallower hydroperiod areas, gluing those mats together, retaining moisture, also a carbon dump in the way that didymo also dumps carbon when it can't do anything with the carbon because it's so phosphorus starved. And finally, just a little story we're learning about the contribution of these diatoms to calcium carbonate formation and the formation of marl soils. You heard about that this morning from Samantha Hormiga, who was able to show from her master's work and her that picture from graduation just this spring. She has this lovely paper already submitted on carbonate sediment production. They build these marl soils, and she's able to show certain diatoms that are taking up bicarbonate, precipitating out calcium carbonate, and in so doing, and building up soil elevation as sea level rises. Very meaningful contribution to resilience as being a part of this very complex, interconnected community. being interconnected. It means it leads to resilience in this ecosystem. And then our ecosystem, right? All these connections are mimicking the connections in our diatom world. Um, and we're all very codependent, just like they are. I wanted to end with a statement that I think this next generation of scientists are much more than scientists, they're much more than diatomists, and they do all kinds of cool things. The one area of connectivity that I didn't really talk about, I talked about connection to each other in our community, connection to place, connection experienced by our study organisms. But we also need to connect with our public as much as we possibly can, as we are trying to restore and maintain these ecosystems and their resilient properties. And the way that we've been trying to do that in our lab is through working with artists and residents. And we've had a number of really lovely artists who have been in and out of our lab. But my lab recently has said, well, we don't really need them. We can do the art ourselves. And I've had the privilege of teaching a course on creative science communication, bringing in some of those artists to help our grad students learn how to do artistic expressions of their work. And so Tommy's written some beautiful poetry about sea level rise and painted really incredible paintings of diatoms. Paige has done the same with her poetry and her incredible depictions of periphyton and of diatoms here. Hannah's done some beautiful stitch work of diatoms, and they've shown this work at exhibits, and they get together on a regular basis to do this kind of thing, and it's pretty cool. Paige even has a musical composition based on her data for marimba that sounds really, really cool. Anyway, that's a little summary, and I just want to really thank my lab who makes life really fun, and the studies that we do really exciting, and do amazing work, and Charlie and this whole community here who have changed the course of both my career and my life. So I'll take any questions that folks might have. Questions about the good old days?
Anyone? You made some comparisons between periphyte and mat communities in Iowa versus periphyte and mat communities in Florida. Now, when you look outside right now, you're not in Florida. So what are the differences between these types of communities? What are the different drivers that are in these two different types of periphyte communities?
Well, I like thinking about what's similar in terms of we have learned that the diversity of diatoms in the Everglades is extremely low, and that these particular diatoms have some strategies for dealing with extreme environmental conditions, really low nutrient concentrations, which is very common in carbonate rich environments, because the carbonate is really scavenging all the phosphorus out of the water, and periodic drying. And so these diatoms are dealing with that where other diatoms can't, or, and so maybe they don't really like it that much, but they can handle it, which was one of the points on the Houston little slide that I showed. And so maybe, the Mastigloia, Grevillei, Lacustris, Calcarea group is able to squeeze out that mucilage. It makes the carbon dump. It makes it nice to persist through drought. It's dealing with very low phosphorus environment. Oh, I forgot to make a point of Katie Stansbury's work, which showed that the halo is also it's bigger when bacteria are present. She added an antibacterial to some of our treatments and showed that the halo shrinks. And So they might be making the mucilage to encourage bacteria who can kick out like alkaline phosphatase and make phosphorus more available to them. So it might be that true collaboration going on. I went back and looked at some of the old slides from Excelsior Fen when I was here in 2015, I think, when you invited me, or Sylvia, and saw that that community has a lot of the same genera, right? As what we see in the Everglades, different species, and the different species are probably because of thermal optima and other kinds of differences in this temperate versus subtropical kind of range. Yeah, but it's similar genera. And I saw that in Canada too, collecting some of the fen-like habitats up there and such.